Hey guys, welcome back to One Stop Biology. So guys, today we are going to start off with the topic cell protein, and then we are going to cover tissue culture, and quickly we'll finish the chapter 9 of class 12 NCERT, which is strategies for enhancement in food production. So let me just remind you that if you have not gone through the previous videos, we have already finished animal breeding and plant breeding together in two videos. So guys, if you have not gone through those videos, please go and check it out and do let me know in feedback, as in, by commenting on all the videos, whether you like it or you want me to change it as per your requirement. Guys, your feedback is really important for me so that I can improvise all these videos. And yes, if you do like it, please do share it with your friends and give it a go ahead by liking the video. And yes, if you're new to the channel, please do subscribe to the channel and press the bell icon for getting all the updates. So guys, let's start off with the single cell protein. Now what happens, guys, that till now we studied about all the conventional method of breeding, right? So these conventional agricultural production of all the cereals, pulses, vegetables or fruits, this might not be able to meet the demand of food at which the population is growing, because the human population is growing at a very faster rate, and to match that it is very difficult to produce all of these vegetables, fruits, or cereals through that conventional way of production. So basically, in order to compensate for that, we have alternate sources of proteins for animal and human nutrition, right? So this alternate source, as I said, alternate source of, say, protein, right? And for whom? For all the animals and humans. This is known as single cell protein. So single cell protein, or SCP, is nothing but an alternate source of protein for all the animals in general, including humans. So what happens in that? So basically what happens is that the microbes are grown on an industrial scale. That means microbes are grown on a very large scale as source of good protein. What happens? Microbes are grown in a very large scale as source of good protein, right? So say, now we know that uh, blue green algae like spirulina, that can be very easily grown on materials like wastewater, from any plant, any processing plant that contains starch, like potato processing plant, or straw, or molasses, or animal manure, or even sewage. So if we produce large quantities of spirulina and we serve it as food, which is rich in protein, minerals, fats, carbohydrates, and vitamins, right? So what we are doing basically is we are decreasing the dependency of a population, on cereals or pulses or vegetables or things like that, right? So basically, such utilization not just compensates for the demand of food, but it also reduces environmental pollution. So there are many microbes like that, right? So for example, bacterial species like Methylotropus, right? So basically, because of that particular bacteria is known for its high rate of biomass production and growth. So basically, that can be expected to produce almost 25 tons of proteins, right? In the same way, we have edible mushrooms, right? So a lot of us, even including me, I like mushroom a lot. So we all eat edible mushroom, right? And we know for the fact that mushroom production can be scaled to a very good extent. So say large scale production of mushroom, large scale mushroom culture is, basically doing what? It is compensating for the regular staple food, like, or vegetables or fruits, and it is providing a food, right? So basically, that is why we say that mushroom culture is a very growing industry. So guys, this is all about the single cell protein. Now let's see what exactly is tissue culture. So guys, tissue culture. So what happened during 1950s is that, scientists somehow experienced that, whole plants could be regenerated from a very small part of a plant which is taken out and it is grown in a test tube. So this part is known as explant. It is known as explant. So basically, what these scientists discovered is that from this explant, one can easily grow the entire plant, right? And so basically, these explants are taken out and they are grown in a test tube. And then after some time, they are again transferred to the normal soil, right? So basically, guys, this this tissue culture is nothing but the same thing, So basically, the capacity to generate a whole plant from any cell or explant is nothing but totipotency. It is known as totipotency, right? I'll repeat it again. Capacity to generate a whole plant from any cell or explant, any part of the plant is totipotency. It is totipotency, right? So basically, what is happening is that you are taking the explant, and what you are doing is you are providing a medium which is nutrient rich, right? And it is happening in a test tube. So you are providing a nutrient rich medium. Say it has the sucrose, it has all the vitamins, it has all the amino acids, it has all the growth, like plant growth regulators like auxins, cytokinins, and all that, right? And you then place the explant in that medium in a test tube, right? And now, what is happening is basically by this method, what you can do is that in very less space, you can propagate a large number of plants in a very short duration. So basically, this method of producing thousands of plants through tissue culture is known as micropropagation. So basically, micropropagation is what? Method of producing thousands of plants through tissue culture is known as what? It is known as micropropagation, right? So now what is happening is that in here that each of these plants, they will be genetically identical to the original plant because you are basically doing nothing but taking out a part, a tissue, and culturing it, right? So the plants that are grown, which are now, the progeny plants basically, right? These plants will be genetically identical to the original plant from which they are grown. And that is why these plants are known as soma clones. They are known as soma clones, right? So there are many plants like that, food plants like tomato, banana, apple. They all have been produced on commercial scale using this method, right? Now, what is another important application of tissue culture? So basically, another important application of this method is recovery of healthy plants from the diseased plant. Now, say that, if a plant is infected with a virus, right? The apical and axillary part of it, the meristem of that plant is basically free of virus, right? So what I can do is that I can remove that meristem and I can grow it in a test tube. I can grow it in vitro and I can obtain a virus-free plant, right? So in the same way, for banana, for sugarcane, for potato, scientists have cultured meristems from diseased plants, right? So this is another, a plus point in application of tissue culture. So now, guys, basically apart from this, there is one more very important and very, very interesting thing that one can do. So basically producing hybrids, right? So one can also easily produce hybrids through tissue culture. What one will have to do is that you will have to isolate. So what you have to do is you isolate single cells, right? And how do you isolate it? You digest their cell wall, right? And then isolate the naked protoplast, right? Now, what you do is that you take isolated protoplast, say from two plants, right? Varieties of plants, which you want to make hybrid of, right? So you take these two varieties, right? And then you fuse both the protoplasts and you get the hybrid plant, right? So basically what you are doing is you are taking two different, you are taking two plants, you are isolating two single cells. Basically, you are digesting their cell wall and then you take out the naked protoplasm. Now you isolate from both the varieties of plants and you fuse both the protoplast and they become a hybrid plant. These hybrids are known as somatic hybrids. Why so? Somatic is basically all about cells apart from, the genetic portion, right? So these basically what you're doing is you are fusing the protoplast, right? So that is why it is known as somatic hybrids. And this process again is known as somatic hybridization. This process is known as somatic hybridization, right? So basically, now imagine a situation where protoplast of tomato is fused with that of potato and then that is grown. So now this new hybrid plant will have tomato and potato both characteristic, right? But this will not have desired combination of characteristics for its commercial utilization. So what does it mean basically? It means that hybrids are not made randomly. Hybrids are only made with plants, for which there is a commercial market. So when there is a demand for such hybrids, then only we make these hybrids. We do not just, for the sake of making hybrids, we do not start making random hybrids. No one would require a hybrid of tomato and potato, right? These are two different, very different food items, right? So always these hybrids are made as per the demand the commercial utilization of that hybrid, right? So guys, with this we have finished single cell protein and tissue culture, and I hope you would have understood it well. So please do not forget to like it, share it with your friends, and yes, I'll repeat again, if you're new to the channel, please subscribe to the channel and press the bell icon. And guys, since we have also finished another chapter, which is chapter 9 for class 12th NCERT, I hope you would have be having doubt. It's good to have doubts. That means you are able to understand some parts of the chapter. So yes, please do let me know in comments for these videos if you have any doubt, and I'll clear it off as soon as possible. Or you can also message me on the WhatsApp number and ask your doubts. The WhatsApp number is given in the description of the video. Okay, guys, see you in the next video with a very new important chapter. Take care. Bye bye.